Please welcome Hilary. She's a computational biologist. She's a faculty member at a number of places, so let me just try to get this correct. Harvard Medical School and a co-director of the program in medical and population genetics at the Broad. And I think I missed the Mass General Hospital as well, but okay, so let me continue. So her group combines genetic data with molecular data to understand the origins and mechanisms of disease. She holds a bachelor's from Harvard in mathematics and a master's from the Weizmann Institute in theoretical computer science. Very interesting background. In particular, she worked on geometric group theory for master's. And then she joined MIT for a PhD in applied mathematics, and her work was on statistical genetics at the lab of Alkis Price at Harvard School of Public Health. And she has numerous awards such as the Hertz Foundation Fellowship and NIH Director's Early Independence Award, but I will not name the rest and let her speak. Please welcome.
Great. Thanks very much for that very nice introduction. And thanks for the invitation. I'm excited to be speaking here. As I mentioned earlier, I am now, I'm going to talk about work in statistical genetics, and I hope that I've included appropriate background. But if there are any parts of it that aren't clear at any point, then I hope that you'll stop me with questions, and I think I'll be able to monitor the chat as I'm going. So I want to start by talking about genome-wide association studies. So in a GWAS, we typically have some people with a disease and then some healthy controls. And if we look at their genomes, which we can visualize like this, we can imagine that this person at this particular location has an A and a T from their two parents, while this person has an A and an A. And so we can genotype these individuals and then look, first of all, orders of magnitude, we can genotype these individuals at between 10 to the 5, 10 to the 7 common SNPs, as these columns are called. And then we're typically looking at data for between 10 to the 4 and 10 to the 6 individuals. And now what we can do is ask at each of these locations whether the genotype correlates with the phenotype. In this case, our outcome is disease status, case control status. So, in this cartoon example, we can say whether you have an, 1 or 2 or 0 A's here doesn't really correlate with whether you're a case or control. But if you look over here, it looks like A's are more common in the controls and C's are more common in the cases. And so if we just do logistic regression or some other type of basic linear model, then we'll find a p-value for association between this SNP and our disease. So in a typical GWAS, we're going one at a time, and for each SNP we're asking, is this SNP correlated with our outcome? So here's some real data. Each dot here is an individual SNP. And then the y-axis is showing the significance of association with schizophrenia in this case. So higher is more significant because this is on the -log10p scale. And after correcting for multiple testing, our threshold for significance is shown by this red line. And what you can see is that there's over 100 genomic locations or loci where there are SNPs that pass this genome-wide significance threshold. And so to take a step back, this is very exciting for those of us who study GWAS. We see each one of these hits is showing that there's a particular genomic change that's correlated with disease. And if we've appropriately corrected for confounding, then that means that there's a variant that's close to, that there's a variant in this region that causally influences risk for disease. And so this thing where, your disease status can't change your germline genetics means that here we've got these windows into the potential molecular mechanisms of disease. But if we zoom in, we see that it's actually really hard to interpret these. And it's really hard to go from here's, at chromosome 1, 2 million base pairs, there's an association with disease. It's hard to go from that to actual statements about what's causing disease. And so if we zoom in on one of the loci, here's the kind of pattern that we might see. So here again, every dot is a SNP and the y-axis is -log10p, but you can see that there's not just one SNP that is highly associated while nothing else is. Instead, there's lots of correlations among nearby SNPs. And so if there's a causal variant somewhere here, then we'll see many associations of many different SNPs, all of which are correlated with that one causal variant whose identity we might not know and who might not have been included in our study to begin with. In addition, even if we knew exactly what the causal SNP was or the causal genetic variant was, that doesn't tell us about genes. And so here you can see each dot is a SNP, and now these are where the genes are in this locus with the red diamonds showing the transcription start site. And so what we'd really like to do is to be able to nominate, okay, first of all, what we hypothesize is that there's often one or more genes that mediates the association at the locus. And so what that means is we think that there's a causal variant, that it causes disease or contributes causally to disease, and that it does so via affecting either the protein coding sequence or the expression level of one of the genes in or near this locus. And so the problem is it's how do we say, oh, it looks like it's this gene, or it looks like these two genes, or it looks like really there's no plausible candidates in this locus. This is very important because if we can name a gene instead of a locus or instead of a variant, then for example, we can perturb it in experimental systems. We can look for orthologs in model organisms. That gives us a much better handle to study biology than looking at this pattern of correlations and trying to say something. So the problem that I'm going to talk about today is how do we take this data and then incorporate other types of data to nominate individual genes at GWAS loci. And before I continue, maybe I'll ask, are there questions so far? Okay. So this is, of course, an extremely well-studied problem. And I'm not going to go in depth on all of the different ways that people have thought of, but I just want to give you a sketch for the kinds of ways that some people have thought about this problem. So one class of methods for nominating genes at GWAS loci is what I'll call locus-based methods. So this includes many different methods. One would be, first, you nominate a variant at the locus, and I'll talk about that later. And then you can ask, is this a coding variant? Does it actually fall in the gene and change the protein coding sequence of the gene? If so, that's super strong support for that particular gene. But this happens a minority of the time. Another possibility is to look at local epigenetic signal. So again, maybe we start by nominating a particular variant, and then we can ask, do we have evidence that this variant regulates a gene? And this comes from totally orthogonal data. For example, for those of you who are familiar with high C data, we might ask, does this variant fall close to the promoter of the gene in 3D space once the DNA is folded? Or we could ask, looking at certain measures of enhancer activity. Actually, maybe I won't go into that because it's not important for the talk and it gets a little bit jargony. But there's a couple of different ways that we might ask questions having to do with epigenetics and gene regulation that could help us predict for this variant which genes might it be tied to, and then we can nominate genes in the locus on that basis. Another type of data that people use is EQTL data, which is another type of data that can help us understand which variants regulate which genes, and which can help us, once we've nominated a variant, start to look at which genes might be affected by that variant. And then there's super simple ways, like saying, I'm looking at my locus, I've nominated a variant, it's probably the closest gene. I don't really understand why, but it seems like variants are more likely to regulate genes that are close by than far away. As a spoiler alert, that turns out to actually be like a pretty good way, like that has a pretty good accuracy in a lot of the benchmarks that we've looked at. So all of these are ways of saying, okay, I did my GWAS, I looked across the whole genome. I identified one particular region of the genome where a lot of different genetic variants are correlated with my outcome. And now I went in and I dissected and I tried to understand what's going on there, which variants are driving the association and which genes do they regulate. But there's another way and there are a large number of methods that go about this type of analysis. But there's another type of approach that's been taken by a couple of groups, which we call similarity-based. And the idea there is that you have many different loci all across the genome that all harbor these disease associations. And we might imagine that if there are causal genes in these loci, they share some properties. And so maybe causal genes in different genomes, in different loci across the genome fall in similar pathways or are expressed in similar cell types or are neighbors in some kind of network. And so as a cartoon, you could imagine, I'm gonna look at my genome-wide GWAS data. I'm gonna find out that genes that are expressed in red blood cell progenitors tend to fall in my genome-wide significant loci. Then I'm going to look at the genes in my actual locus itself, and I'm going to say, hey, look, this gene is expressed in red blood cell progenitors. It looks like it might be the kind of gene that we see associated with this disease. And there are a lot of different ways to formalize this, and it's been kind of instantiated using different data and different methods in a couple of ways in the past. And so can
I ask a quick question, Hilary? I must have missed this, but how what is the order of magnitude of candidate genes per identified locus?
Oh yeah, depends on how you define the loci, but single or double digits. It would be a big locus if it had more than 20 genes.
Okay, thanks.
Yeah, so here we go. We've narrowed it down to, I don't know, order of a dozen genes. We want to be nominating probably one of those genes, but could be 0, could be more. This is a fundamental problem. There's many different ways that people have solved it in the past, but we felt like information was being left on the table. And for something of this level of importance, we should really be using all of the tools that we've got. So one area that we focused on was thinking specifically about similarity-based gene prioritization. Can we set up a framework for doing this that allows us to take advantage of many sources of information about genes? And so what we want to do is build a framework where not just as new data sets are generated, but also as new data modalities are developed, that as long as we can encode things as a piece of information about genes, a vector that's got one number per gene, we want to be able to take advantage of that information to prioritize genes for disease. And the second thing that we wanted to do was not impose this threshold and then look at things that pass or don't pass the threshold, but rather do some genome-wide modeling. And I, in my PhD, did some work that showed that if you use statistical methods that leverage information from the whole genome instead of just restricting to genome-wide significant loci, that can really increase the signal that you see and improve our ability to understand disease biology from these data. So then also for gene prioritization, our goal is not to develop one method that just answers the problem, but instead what we want to do is take advantage of the fact that we've got these two different ways of looking at the same problem. So we want to be able to combine the locus-based approaches that are, carefully dissect which variants regulate which genes. And we want to combine those with these similarity-based approaches where you're looking at genome-wide patterns of enrichment for pathways and networks and expression. We'd like to combine those types of information. And the with principled evaluation part is one that I'll really focus on. So one theme in this project, but also I would argue in computational biology more generally, is how can you tell whether you're doing well or not? And that can be very difficult. And so I'll come back many times to emphasize how we thought carefully about how to tell whether what we're doing is working or not. So today, I will describe our new similarity-based method, which we call polygenic priority score. And then in the second half, I'll talk about how we combined POPs, as we call it, with locus-based methods, thought carefully about evaluation and then nominated specific genes. And this project was led by Ellie Weeks in my group. So the intuition behind our POPs method is that we have lots of different kinds of information about genes. So we can ask for any gene. Is it expressed in excitatory neurons? Is it a synaptic gene? Is it a protein-protein interaction neighbor with some other gene? And so for each of these gene features, as we're calling them, so a gene feature, one number per gene, we can ask overall for schizophrenia, given this GWAS data, is it enriched? So, we've seen that excitatory genes that are highly expressed in excitatory neurons are enriched in some sense, and I'll come back to exactly what we mean by that, are enriched for a GWAS signal. But genes that are highly expressed in epithelial cells are not enriched for schizophrenia GWAS signal. And now, if we look at our particular 21 genes or in our locus, then we can ask for each gene, does it have these features that are enriched for our phenotype? Is it highly expressed in neurons? Is it a PPI neighbor of gene Y? And then if we see a gene that seems to match up really nicely with our genome-wide enrichments, then we can prioritize it. So that's kind of the cartoon version of what we're doing. For this audience, you may be more interested in how did we actually set up the problem. So the first thing that we did was take all of the different information and data sets that we have about genes and encode it in a gene feature matrix. And so this is kind of the modular part where we want to first take network data and featureize it, take pathways, binarize them, take expression data, do some kind of feature extraction. And then once we've done all of that, we have one big matrix. The rows are genes, the columns are gene features. And so the problem that we're trying to solve is if we break off one of these rows, we want to know, does the gene corresponding to this row look like it should be a disease gene? So what kind of function of gene features will give us a score that tells us something about whether a gene is likely to be a disease gene? So how can we find this function? Ideally, what we would have is a very large set of known disease genes and known non-disease genes. And if we had such a very large set, then we could train a predictor using all of these features. That would give us F, and then we could apply F to any gene that we wanted. But we don't have these gold standard data, and certainly not we don't have it for many genes, and we don't have it for many phenotypes. And so instead what we do is we're using noisy training data. So we have some kind of score that we think genes that have high scores are more likely to be disease genes, genes that have low scores are less likely to be disease genes. And if we can have this score for many, many genes, then that should give us some information that we can use to train our function with. And so where do we get this score from? Well, now we're going to go back and use our GWAS data. So we can say genes that fall in associated loci or that have SNPs nearby that are associated with our phenotype, those are more likely to be disease genes. And this is very noisy, of course. Like I just said, if you fall in a locus, you might be one of 10 genes that falls in the locus. But overall, genes that are near GWAS associations are enriched for disease genes, and genes that are very far away from any GWAS association are depleted for disease genes. And so with this GWAS data, we can take advantage of the fact that we have it for all of the genes in the genome, 18,000 protein coding genes. And then we can use this to train our predictor. So we
quick fine point question. Did you consider using fine mapping data for that step?
Yeah, so that's what we started with, and it turned out to not work very well. And I think that part of the problem was that's limiting to genome-wide significant loci again. And so, or at least if you only apply the fine mapping to the genome-wide significant loci.
I see, right. And here you want to just kind of give a score, and it's sort of just how much significance is there in that genic region overall.
Exactly. I think that there's a trade-off between,
yeah,
I guess one way to think about it would be like noise in the outcome versus number of observations.
Yes.
And so, especially for polygenic phenotypes, I guess one theme that has arisen for us in our study of polygenic phenotypes is having information for 18,000 genes is a lot better than having information on 200 genes.
Awesome, that makes sense, thanks.
I do think, I'm going to describe our way of solving this. I think that this is still pretty open. I don't think we by any means just like found the way to do this, but we found a way that seems to work pretty well, and I'm, we're excited about extending and improving it in several different ways. So We are using a linear predictor, and the outcome that we're using, we are just taking from this previously published method, MAGMA. So the MAGMA score for a gene is the mean chi-squared association statistic taken across all SNPs near the gene, and then fed through a transformation that gives something that's roughly on a scale, on a z-score scale. So on a scale that makes sense for linear modeling. It's not a it's not derived from a generative model of how the phenotype arose from the genetic variance. And so, but it is a score that's higher for genes that are near highly significant SNPs, and that in previous work has been shown to give good results for enrichment. So previously, when these MAGMA scores were used to identify individual pathways and cell types that were enriched for GWAS signal, it seemed to be doing a really good job. And so, so we built on that previous work that had been shown to work well for pathway enrichment to build our kind of broader gene prioritization model. So we're modeling these as linear combination of our gene feature matrix. We do marginal feature selection and only put features in X that are associated to Y at p less than 0.05. We have a coefficient vector, which is what we're trying to estimate. And then we have a residual that has this covariance matrix R, which reflects the correlation between nearby SNPs in the genome. So the MAGMA scores, because they're derived from SNPs, if you have two genes that are nearby, you'll be using SNPs that are nearby to score this gene and to score this gene, and these SNPs are correlated with each other. So that's put into the residual. So, so I think some features, sorry, to finish describing this, we then fit this with generalized least squares, L2 regularization, because even after marginal feature selection, we still have a couple thousand features. And then we score the genes using our estimated B hat vector. So I think some features of this model are A, we get to use all of the genes. B, we're scoring genes based on nearby association in a way that in general is higher for genes that are near more associated SNPs and lower for genes that are further away from associated SNPs. We've incorporated LD here, and now we've just reduced it to a problem of very high dimensional linear regression, which we're fitting here using L2 regularization and marginal feature selection.
Can I ask a couple of questions? So what's the dimension here?
57,000 in the before marginal feature selection, and I think between like 3,000 and 5,000 maybe after marginal feature selection. It depends a lot on the data.
So
yeah.
For this method to work, you need that each gene feature has enough variability at each locus that you're looking at, right? Because if all the genes at the same locus have exactly the same gene feature vector, then you're just going to pick one at random with this procedure.
Yeah, that's right. That's right. So a lot of the features are, so there's a combination, well, maybe we can come back later to what exactly the features are, but that hasn't been a problem that we've run across where all the genes get the same score.
Okay.
But maybe it would make more sense to come back to that when I'm telling you exactly what we put in the feature matrix.
Okay.
Great. Before we get to that, I just want to mention for this audience that we considered some alternatives. Marginal feature selection and L2 regularization seems, that's not what I expected to do the best, but it did much better than just throwing everything in and using L1 regularization. It did better than marginal feature selection followed by L1 regularization or followed by no regularization at all. And surprisingly to me, at least, GLS versus OLS did not make as big of a difference as we thought it might. So you can actually just neglect the correlation among the gene scores and you don't get too much of a hit in our current version of the method.
So sorry, go ahead, Sasha.
What was on the y-axis in the previous?
Oh, good point. I should have So later I'll describe the different ways we have for evaluating how we're doing. So this is a method called benchmarker. And the idea is if you use information on 21 chromosomes to nominate genes on the 22nd chromosome, then you can ask whether the genes that you're nominating explain more heritability, whether variants near those genes explain more heritability than random genes. And so this comes with a caveat that I, again, will describe in a later slide, but the idea is to what extent, if we can't see the GWAS data ahead of time, are we telling you already which genes tend to fall near associated SNPs and which genes don't?
Thank you.
So my question, so I'm not sure what the magma score is exactly, but you said it was like some sort of chi-square score, so I'm presuming this is a positive number?
So what they do is they take the average chi-square statistic of variance in and near the gene, and then they compute from that a p-value that takes into account the fact that the variants were correlated, that were used to compute that mean chi-squared. So they get some null distribution of what an average of correlated chi-square statistics is. And then they go back and z-score transform that p-value. So they just, inverse normal transform the p-value. And so what that means is that you do get both positive and negative scores. And they're not actually normally distributed across all genes, because to the extent that you had significantly associated genes, you had more significant more significant p-values, which meant z-scores that are going to be higher. But they transform these gene p-values to z-scores.
Okay, so in a way, it's some sort of a link function that you're applying, but you're just like folding the noise into it somehow, right? Like it sort of is a generalized linear model that you actually, right? So given like generalized linear models, they're just a way for you to actually map x beta to whatever range your conditional expectation should actually live in. And here it should be positive, and the way they actually do it is by just applying this like z transform to it.
Yeah, there's two steps, because the step to go from mean chi-squared to p takes into account correlation, which will be different. That you'll be taking an average over different numbers of SNPs for different genes, and the SNPs will have different patterns of correlation. So going from mean chi-squared to p is something that's gene dependent, but then going from p to z, I agree, is just a link function.
Okay, all right, thanks.
So now, Philippe, we can come back to your question about features. So the features that we included were, first of all, pathway features. So these are just yes, no, binary. Are you a synaptic gene? Do you cause, I don't know, kidney function problems in mice when you're knocked out? And then there's protein-protein interaction features. So these are, again, binary. And what we did there was for each gene, we put a 1 for any gene that's a first-degree neighbor of that gene in the PPI, and a 0 for all other genes. And then the tricky part was gene expression-based features. And so this might be one slide of Inside Baseball for people who think about gene expression. But there's lots of information about gene expression in a lot of places. And Jacob and Nathan in the group downloaded 77 publicly available gene expression data sets. Most of these are single-cell RNA-seq from a variety of different tissues and systems, all publicly available. And then to featureize a gene expression data set, what they did was PCA and ICA gene loadings from the whole data set, followed by clustering, followed by average expression and PCA loadings from within PCA, within each cluster. Within cluster PCA, followed by a comparison of each cluster to the union of all other clusters using both a quantitative statistic and then a threshold for differentially expressed genes. And by putting this large number of data sets through this very systematic pipeline, we got another 40,000 features based on gene expression. And these, for the most part, as you can see, are, for example, gene loadings from PCA, or average expression within a cluster. So a lot of these are quantitative. And so now, could we come back to you had a particular question about, you've got, your dozen genes in the locus, and are you worried that you're not going to have enough variation in the features that you've got to differentiate among these genes? And so what we found is that part of that depends on how much of your trait biology is reflected in these features. So for example, something that we found in previous work is that there's often good power to do an enrichment analysis based on GWAS data that tells you, for example, neurons are enriched for schizophrenia GWAS. Genes that are highly expressed in the liver are enriched for LDL signal. Genes that are highly enriched in T cells are enriched for rheumatoid arthritis signal. And so I don't think I can say any numbers about how often are genes in which loci supported by which features. That's something that we're looking into. And there's also a lot of redundancy. You know, we've got these pathways, which then often contain genes that are expressed in particular cell types, or the same cell types are represented in multiple data sets. But as I'll show in some of the examples, it happens pretty often that you'll have 10 genes and then one of them will be a neuron expressed gene, or one of them will be a DNA damage repair gene. And so if your features are doing a good enough job of capturing what's going on, then to the extent that's true, this method, you know, will attempt to pick up on that, and then we'll attempt to score genes. Does that answer your question?
Yeah, I mean, honestly, I'm not even sure. So, you know, one diagnostic that I would try to run probably would look like, you know, just order your genes according to Locus, say, and just like form like a big correlation matrix of those features that you created for all those genes. But the thing is, I'm not even sure whether I would want to see a nice, you know, tapered off thing, or where, you know, I go from the diagonal, those like, or maybe a banded thing, you know, that's kind of what you would expect, that things tend to be more correlated when they're close to the diagonal and less when they're further away. But which would be some sort of a good sanity check. But at the same time, I think I'm trying to argue for the fact that you actually don't want even anything close to the diagonal, right? You want something that just becomes very quickly independent as you go. At the second you walk away from the diagonal, actually, you need very fine drop immediately after you go away from the diagonal, basically.
Yeah. Okay.
And so maybe zooming in on this diagonal would be a good diagnostic to check if you get the variability you're looking for.
Yeah. Okay. So that gets to a really important feature of these data, which, you know, we should quantify this, but overwhelmingly, looking at the data by eye, there's not a lot of autocorrelation across the genome when it comes to these features. So if you look at genes that are highly expressed in neurons, they don't tend to cluster together in the genome. And there are definitely exceptions to that. There are definitely gene families that all are, you know, all right together in the locus and that all fall in the same pathway. But for the most part, if you're looking at, synaptic genes, there's going to be a lot of those scattered on different chromosomes. And when you look at one locus, there'll be, you know, one calcium channel gene next to an immune gene and a gene, you know, so part of the reason that this works is because the correlation that you get from GWAS scores is autocorrelated along the genome very strongly. But the noise that you get in these features, the features are still noisy, but they're just correlated in a different space. They're not noisy along the, you know, they don't have strong autocorrelation across the genome for the most part, with some definite exceptions.
Okay.
So we took these features, we analyzed 113 phenotypes from the UK Biobank and from publicly available summary statistics. And so now let me get at evaluation. So if we're looking at these similarity-based methods, then one trick that we can use to evaluate them is this idea of leave one chromosome out analysis. And so there's one published method that takes advantage of this, is the benchmarker method that I was describing earlier. And so this benchmarker method takes your gene prioritization method, runs it in a leave one chromosome out way. So now you're prioritizing genes on chromosome 1, having not yet given it access to the summary statistics from your GWAS on chromosome 1. And then you, when you're prioritizing genes on chromosome 2, you don't give the method access to the GWAS data on chromosome 2. And so then you can ask how much heritability is captured by SNPs near prioritized genes. And analogously, you can look just at the genome-wide significant loci. And as I mentioned earlier, more often than you would expect by chance, the closest gene to the index SNP is the one that's mediating the association. And so you can ask whether more often than by chance, the gene that you're nominating is the closest gene. And I have to add this caveat, which we found just recently, which is that while we ran our method fitting B in a leave one chromosome out way, we didn't do our feature selection in a leave one chromosome out way. So that puts an asterisk on the results that I'm going to show with updated results coming soon. So we compared our method POPs to two competing methods, DEPICT and GWAS. And so this is showing normalized tau is a quantification of the heritability enrichment of SNPs near the genes. And POPs has a much higher, so higher is better by this metric. And POPs has higher performance than DEPICT and GWAS, both on average across the 113 phenotypes and also within these categories of phenotypes. If you take the phenotypes and you classify them into different categories, then it looked like POPs was outperforming other methods pretty consistently across these categories. And there was a similar message if you look at the closest gene metric. We then wanted to ask, as we're prioritizing genes for these different phenotypes, can we look at feature importance? Can we go back and ask, of all of these features that we put in our matrix, which ones are the important ones? So one way that you can ask that question is to say we've got different sources of features from expression, from pathway databases, and from PPI, protein-protein interaction networks. And on average across phenotypes, it is better to put in all of your features than to put in just one type of feature. And it looks like expression and pathway features are on average more informative than the PPI features. In this case, there's more noise and heterogeneity within each of the phenotype categories. And then we can also look within the expression data about bulk expression versus single cell. And it looks like for the most part, using only the single cell expression features gives us performance that's comparable to what we get when we put in all of the expression-based features. And this, of course, is a function of which data sets we include and don't include, rather than a definitive statement about technology. But I think that it shows that we're at least able to extract a good amount of information from these single-cell RNA-seq data sets.
Does your penalty, do you modulate your reach penalty depending on what type of feature you're actually putting there, or everybody gets the same penalty?
We looked into that, and if I remember right, we implemented one version of that, found it didn't make a big difference, and then dropped it. So now everybody gets the same penalty. I think that there's definitely room to look into that more. One possibility, I mean, looking at these results that I just showed, for example, it doesn't look like there's a huge amount of heterogeneity in these big classes. And so it could be that there are other ways to classify features that would have, there are pathways relevant to schizophrenia and pathways relevant to rheumatoid arthritis. So maybe there's some other way to think more carefully ahead of time. But in our, in one version of that, we didn't find a difference. So next we want to ask, for a given phenotype, which feature clusters are contributing the most to the prioritization of prioritized genes for this particular phenotype. And on the one hand, interpretation of linear models should be super easy. We have a vector B. But the problem is there's a lot of collinearity in our feature matrix, a lot of highly correlated features, so that our B hat itself can be quite noisy. And so to solve this, we changed the question instead of changing the solution. And we clustered features and then asked for the contribution of each cluster to the prioritized score instead of breaking it down by individual feature. And so now to quantify the informativeness of a feature cluster, we ask, how much does that cluster contribute to the scores of prioritized genes? And everything is linear, so this is super nice and easy. The POP score of gene G is an inner product of the feature values for gene G with the vector B of coefficients. So you can break that as a sum of contributions from these disjoint clusters. And so then you can take this contribution of cluster C to gene G and just sum it across all of the genes that were prioritized. And if you look at the results for phenotypes, so then you have to go into the clusters and manually annotate them, which can be a little tricky. But Jacob and our group went through and did this analysis for several phenotypes and found, for example, clusters related to lipid biosynthesis for LDL, clusters related to chromatin organization, calcium channels for schizophrenia, HbA1c is an interesting phenotype that has a component both of blood cell biology as well as glucose regulation, which were identified by the top 2 feature clusters, and similar immune-related clusters for rheumatoid arthritis. So I'm going to pause here again because we're about to move on to the second half. But if there are any other questions about the POPs method before we talk about locus-based and similarity-based methods, I also see that I don't have a ton of time, so I'll cut some out of the second half of this talk. But I'll pause again for questions so far. Great. Okay. So let's see, is it quarter till? Great. So this idea of combining locus-based and similarity-based methods, right now we're really happy because we've got a method that lets us leverage genome-wide signal. It lets us throw in, tens of thousands of gene features. It lets us extend or contract our gene feature matrix as we would like. And then it gives us a way to score individual genes on the basis of these gene features, which very importantly, as Philippe, you were mentioning, that gives us a noisy score. It's not necessarily better than these scores that nominate variants and then connect them to genes, but the noise is going to have a very different pattern. And so what we want to do in section 2 is to look at how can we combine these 2 very different types of methods, and can we use that in particular to start to nominate genes with higher confidence than what we can do with individual methods. So this I won't go into too much. We ran locus-based methods, but just simple closest gene types of methods, or the magma score that we use to train our predictor, coding variants, as I mentioned earlier, several different types of epigenetic-based methods, and a more stringent and a more lenient EQTL-based method. I won't go into that too much. And I'll also leave out, well, actually, okay, this part is really interesting, I think. So in order, some of these methods require us to nominate variants at a locus. This is something I've alluded to a few times, but then I haven't really talked about how we do it. And so to recap what I said at the beginning of the talk, when you're doing a genome-wide association study, you're doing marginal regression at every variant. So if you imagine that, what we think is going on is that there's a very sparse set of causal variants, like maybe there's 2 causal variants in this locus. But then because everything is correlated with everything else, and because you have finite sample size, you wind up with this, hard to interpret and kind of complex pattern of association that you look at when you're looking at GWAS signals. So why do we do GWAS in this way? I've had this conversation with statisticians a couple of times who wonder, why would you do marginal association to begin with? And it's actually kind of genius. So first of all, it's very robust and very interpretable. But also, we don't want to assume that the causal variant was actually typed. And so it could be that there's some, and this was especially true in the early days of GWAS, it could be that there's some structural variation or some kind of rare variant or something like that. And so if you're doing these marginal associations, then if you are looking at correlation of a variant that tags a nearby causal variant, then you'll be able to see the association even if you didn't necessarily measure the causal variant itself. But also, I think that model misspecification plagues this field pretty seriously when folks develop sophisticated methods. And so having the first line analysis be something that you can do via some inner products is very nice. The con, of course, is that it doesn't nominate candidate causal variants. It just shows you this kind of association of a bunch of variants that are all correlated with each other. And so statistical fine mapping is the process of trying to nominate causal variants. And the state-of-the-art methods for statistical fine mapping are different versions of Bayesian sparse regression. And this is a hard problem. And so, if you want to solve it exactly with a prior that says that actually enforces sparsity, then this is very hard. And so there's been kind of a series of work done developing approximation algorithms that are computationally tractable and that don't lose too much accuracy. And the current state of the art algorithms are based on stochastic search and variational inference. And so the output that you see here is, for example, the posterior inclusion probability is the posterior probability for each variant that it is a causal variant. And then in this example, we've got one SNP that's got a very high PIP close to 1, and then another 5 SNPs that have a lower PIP. And some fine mapping methods also structure the posterior in a way that lets you build credible sets. And so a credible set can be interpreted as a set of variants that with 95% posterior probability harbors a causal variant. So you can say, I know that there's one causal variant that's here, and then there's another causal variant. It's one of these 5. I'm not sure which one it is. So these algorithms can also require some thought to actually apply to your data. And my Masa Kanai and Jacob Jurlich in my group applied these 2 state-of-the-art fine mapping algorithms to a whole bunch of loci from 2 very large biobanks. And that's the set of results that we're going to use. And just to continue on this tangent for one more minute, once we've applied these algorithms, they're misspecified in a dozen different ways that I can tell you. And so one question is, how much are we finding, and how much should we believe it? Can we get any initial sense that what we're finding has any relation to biology at all? And so we found tens of thousands of variants that had posterior inclusion probabilities above 10%, including thousands of variants that have posterior inclusion probability above 90%. And so one way to get at, is there signal in this data when we report these posterior inclusion probabilities, should we believe what we're seeing, is to take the genome and break it down into these categories, which we didn't tell our fine mapping algorithms about, but where we think that SNPs that fall in the exonic category are more likely to be related to disease than unannotated SNPs in this purple other category. And so if you look across the whole genome, you can see that most SNPs are purple, and then there are some that are in these different functional categories. And now if you look at how SNPs are classified when you look in particular bins of posterior inclusion probability, you can see that as our confidence in a SNP being causal increases, then its likelihood of being in these functional annotations increases as well. And so this is one way of using kind of orthogonal data that we didn't give our algorithm access to assess to what extent what we're seeing makes sense. And thinking more about fine mapping methods, ways to benchmark them, methods development in that space is something that I'm very interested in, but that I won't talk about more here. And instead, I'll just mention that we used these statistical fine mapping algorithms to nominate variants from the UK Biobank, which we then could feed into our locus-based methods. So the locus-based methods largely disagree with each other. And I think that maybe right. And so in order to benchmark them, we used a novel set of gold standard genes that allowed us to evaluate both the precision and the recall of locus-based and similarity-based methods simultaneously. And so using this new way of benchmarking, we rechecked that POPs is outperforming DEPICT and GWAS both according to precision and according to recall. And then we could compare all of these different methods. And here, I just, for the sake of time, because I also want to leave time for questions, I think I'll just go through this. I won't go through this in too much detail. But the punchline that we found was that combining POPs together with locus-based methods, which is shown with these boxes, gives us higher precision, higher confidence in the genes that we're actually nominating than using only the locus-based methods by themselves or combining signal from different locus-based methods. And so just to give some examples of results and also to give you a sense of where we're at with this problem at this point. So according to our benchmarking, if you do this kind of stringent thing of you require a gene to have support from POPs and you also require it to have support from at least one locus-based method, that gets us up to the very impressive and great precision of 76 or 77%. And so the idea that at a locus with, so it's still very much an unsolved problem because really what we'd like to be doing is getting up to precision that's close to 100%. But if we look at these genes that we're nominating using multiple different lines of evidence and we examine them, for example, for schizophrenia, DRD2 is a known drug target for most antipsychotics. And then we've got 2 calcium channel genes, so that calcium channel genes are known to be important for schizophrenia and show up repeatedly in different types of analysis. BCL11A is a known intellectual disability gene, and then STAG1 is a gene that just recently was nominated using orthogonal genetic data, rare variant data from exome sequencing at an FTR of 5%. And so we see that as like a pretty strong set of, top 5 results by POPS score. I think I won't walk through some other examples of how we think it's good to use POPS. And instead, what I'll do is thank Ellie, Jacob, and Nathan, who really led this work, and then to mention, as well as our collaborators, funding sources of data. And if anybody's interested, we have a preprint software and data available. And now I'm happy to take any final questions.